Professor SNAB, where we make A-level biology as simple as a cell. So today we're looking at understanding the process of contraction of skeletal muscle in terms of the sliding filament theory, including the role of actin, myosin, troponin, tropomyosin, calcium ions, ATP, and ATPase. So first, we'll start off by looking at the structure of skeletal muscles. So skeletal muscles are organized as follows. So we start off with the whole muscle, which then further divides into bundles of muscle fibers, and then further divides into myofibrils, then into sarcomeres and protein filaments. And I would recommend you kind of draw this out as a flow chart to help you visualize this. Because that's a really good tip actually, because it's going to help you understand the different things we'll look into in a minute, like the sliding filament theory and how the muscle contracts and relaxes. They'll help you visualize how the whole thing works. So skeletal muscle consists of bundles of muscle fibers, and each muscle fiber is a single elongated cell, multinucleate, which means it contains multiple nuclei, surrounded by a cell surface membrane called the sarcolemma. Muscle fibers are formed during the embryonic development by fusion of many cells, and multiple nuclei are needed because the cells are extremely long and metabolically active, which means a single nuclei won't be enough in skeletal muscles. So now to look at the internal structure. So the sarcoplasm contains lots of mitochondria to help with the high demand for ATP production. It also acts as a store for glycogen, and it also has myofibrils. So myofibrils run the length of the muscle fiber, and they consist of repeating units called sarcomeres. Sarcomeres are the basic contractile units of muscle. So the thin filaments are mainly made of actin and associated regulatory proteins, troponin and tropomyosin. The thick filaments are mainly made of myosin. So myosin filaments have globular heads capable of binding to actin and hydrolyzing ATP using ATPase. The striped appearance of skeletal muscle is caused by the arrangement of actin and myosin filaments. So the I band only contains actin and it appears light. So try to think of this in the context of your exam papers, because exam papers are, well, as you would know, it would be usually black and white. But when you do study the topic, use the pictures in your textbook to understand it. So the A band is a mix of actin and myosins, so they appear dark. And the H zone is just myosin, so it appears intermediate. So the sliding filament theory. So muscle contraction occurs because actin filaments slide over the myosin filaments. This leads to the shortening of the sarcomere, and without the filaments themselves becoming shorter. So as the sarcomere shortens, the myofibrils shorten, which leads to the muscle fibers shortening, and then the whole muscle contracts. So now to look at the role of calcium ions in the sliding filament theory. So looking at the stimulation of muscles, when a motor neuron stimulates a muscle, an action potential reaches the neuromuscular junction. The calcium ions are released from the sarcoplasmic reticulum. So what's the sarcoplasmic reticulum? It is a specialized form of the endoplasmic reticulum, and it surrounds the myofibrils and stores the calcium ions. So the calcium ions, which are now released, attaches to the troponin of the actin filament, and this causes the tropomyosin to move. Tropomyosin shifts away from the myosin binding sites on the actin, and this allows myosin heads to attach to actin. So formation of cross bridges. So myosin heads bind to the exposed binding sites on actin, and this is what forms the cross bridges. At this stage, the ADP and the phosphate are attached to the myosin head. So after binding, the ADP and the phosphate are released, and the myosin head changes shape. So what happens as a result? The myosin head pivots forward, and this pulls the actin filament across the myosin filament, and this movement is called the power stroke. The power stroke is the collective action of many myosin heads, which slides the actin filaments inward, and this shortens the sarcomere and produces muscle contraction. So now to look at the role of ATP and ATPase. So the first off is the detachment of myosin. A new ATP molecule binds to the myosin head, and this causes the myosin head to detach from actin. Without ATP, the cross bridges cannot detach. The myosin head contains the enzyme ATPase. So it is the ATPase that hydrolyzes the ATP to produce ADP and the phosphate. Energy released changes the shape of the myosin head and returns to its upright, energized position. The myosin head is now ready to bind again, and the entire cycle repeats. So when the simulation stops, the calcium ions are actively pumped back into the sarcoplasmic reticulum using ATP. So now basically the reverse is happening. So as the calcium ions concentration falls, the calcium dissociates from troponin. Tropomyosin moves back over the binding sites on actin, the cross bridge formation stops, and the muscle relaxes. So rigor mortis is something we've looked at in topic 6. So if you want a revision of topic 6, the entire playlist is already uploaded, so do check it out. So what happens after that? ATP production, unsurprisingly, stops. And without ATP, the myosin heads cannot detach from the actin, and the cross bridges remain permanently attached, as we just looked at right now. And this results in the muscles becoming rigid. And this state is called rigor mortis. So that is all for Professor Snap today. Hope you found this video useful. If you have any questions, let me know in the comments below and I'll get back to them as soon as possible. And don't forget to like, share,